Days to everybody. I'm so glad you could join us. I'm Linnea Fletcher, Principal Investigator, Executive Director for InnovATEBIO, the National Biotechnology Education Center. And this is part 2 for our PCR webinar series. Part 1 has already been recorded and is available. Cassandra will share that with you. Cassandra Mitchell-Nuthouse is our grant coordinator. And I want you to remember, besides the 3-part series for PCR, we also are going to have a workshop in July. You'll need to save that time period. It'll be face-to-face at St. Louis, just like it was this past summer. I'll now introduce you to Bryony, who's head of education for Bio-Rad, who has graciously helped, well, let's say organized this PCR webinar series with InnovATEBIO. Take it away, and thank you.
Thank you, Linnea. And thanks to the InnovATEBIO team and St. Louis Community College, and in particular, our speakers today for helping to bring these technologies to our community. We obviously are not going to cover everything in advanced PCR technology today, but I want to make you aware of some resources that are available to you. So we just had a webinar 2 weeks ago, but we also have had previous webinars in advanced PCR techniques in collaboration with InnovATEBIO and St. Louis Community College, as well as others. Those are available online. And we have Bio-Rad bulletins on teaching real-time PCR that covers things like instruments and reagents, as well as droplet digital PCR. The Explorer program from Bio-Rad has 2 teaching kits, hands-on kits, reagent kits, to help simplify bringing real-time PCR to students. And lastly, Bio-Rad Life Science Group has free and full online courses, like 5 or more hours of content for researchers on real-time and on droplet digital PCR as part of Bio-Rad's Academy. These go really deeply into the technology, how to set up assays, how to do experiments, how to analyze data. So if you want to get really deep into this, there are resources there. And we're going to send you information on those resources after the webinar, when we send you the slides and the decks and everything. So on today's agenda, we have 2 guest speakers, and we'll have a short Q&A after each speaker, and then we'll have a facilitated discussion on teaching advanced PCR to students. We have Andy, who's going to give us a primer on PCR technologies, and Claire from Andelyn Biosciences, who's going to tell us how she uses advanced PCR in her role as in analytical development. We also have Maranda Thanasiu from Andelyn's talent development team here, who can talk to us about careers in CDMOs, contract development and manufacturing organizations. And I'm going to formally introduce Andy and Claire before their presentations. Our facilitators are Beth Elam Mchaud and Damon Tighe. Beth is a business specialist for the BioBench Contract Research Organization at St. Louis Community College's Center for Plant and Life Science. Beth works with program students moving into their internships, as well as helping companies that are mostly startups gain access to the center's instrumentation bench space and interns. And I want to note that Beth and her colleague Betsy Bodecker will host next year's hands-on advanced PCR workshop that InnovATEBIO is putting on, that Linnea just mentioned. Damon Tighe is a curriculum training specialist with Bio-Rad's Explorer program. And he's been supporting educators in implementation of PCR projects for over a decade. So Damon and Beth will lead the Q&A after the speakers, and then they'll lead this discussion on strategies to introduce these applications to students. So please add any questions or discussion points that you have into the chat as we progress through the meeting and through the morning. So I would like to introduce Dr. Andrew Prantner. Dr. Prantner is the field marketing specialist in the digital biology group at Bio-Rad, where he is involved in application development and support of droplet digital PCR. He is a multidisciplinary research scientist with experience in biochemistry, biophysics, cell biology, chemistry, immunology, material science, molecular biology, radiology, and reproductive biology. So if you have any questions, just ask Andy. He received his PhD in biochemistry from Washington University in St. Louis, working on the MRI of small animals, and his master's in chemistry from the University of Pennsylvania, working on solid-state NMR of membrane proteins, and his bachelor's in biochemistry from the University of Wisconsin-Madison, where his undergraduate work focused on NMR of soluble proteins. And he's been with Bio-Rad since 2019. Take it away, Andy. Thank you.
Perfect. Thank you very much. Yeah, I guess I've been avoiding getting a real job, just kind of bouncing around, and eventually it caught up with me. Let me share my screen. All right, perfect. So I am going to start by talking about the background on 3 generations of PCR. The 3 generations are what I've been calling a first generation, which is conventional PCR. That most people are familiar with. That's when Taq polymerase and thermal cyclers came online. The second generation is qPCR, which evolved with real-time detection and probe-based assays. And then the third generation is droplet digital PCR, which then started using sample partitioning and Poisson statistics to get concentration. So I'll go through each of these briefly as we go. So conventional PCR, a piece of DNA is just amplified by repeated cycles of denaturation, primer annealing, and extension, right? And the basic component of these reactions is a forward and reverse primer. And the annealing temperature and extension here is shown at 2 different temperatures, which is considered a 3-step PCR, but they can be at the same temperature, and that's what people consider a 2-step PCR. So the PCR, in theory, you get a doubling of the target with each cycle. So, you go from 1 to 2 to 4 to 8 to 16. But, in the real world, experimentally, that doesn't really happen. The actual amplification falls off at later cycles for various reasons. Depletion of reactants. It is a chemical reaction after all. So you start running out of reactants, and then you also get some competition of the PCR products start to self-anneal, and they compete with primer binding, and they prevent that amplification. So you don't get the theoretical doubling, but you do get experimentally, a large amplification as you PCR amplify. So conventional PCR, you sample the endpoint, right, 35 to 40 cycles, and then you evaluate your product by agarose gel electrophoresis, which separates based on size, and it typically has a fluorescent dye of some sort already in the agarose, and that increases fluorescence when bound to double-strand DNA. If you want to get more out of a reaction and look at multiple targets, multiplex it, and you can look at multiple targets. This involves using separate forward and reverse primer pairs to get different size PCR products. On the bottom of this slide, I show a singleplex version of some data I found online, and then a triplex where they put all 3 together and they see, good separation based on size. So that's conventional PCR. Quantitative PCR, or could be qPCR or sometimes real-time PCR, is similar, right? It uses 2 different detection chemistries. One is dye-based, which is completely analogous to the conventional PCR, where you have a dye that gets into double-stranded DNA and the fluorescence increases. You can also use probe-based, which uses, in addition to a forward and reverse primer, a fluorescent probe that's typically got a reporter on one end and a quencher on the other end, so that it's at baseline a very low fluorescence. And then during the extension cycle, as Taq polymerase comes across, its nuclease activity separates the fluorescence reporter and quencher, causing an increase in fluorescence with each additional cycle. And so this is taken, advantage of in qPCR, where the data is an amplification curve, and it's just you'll typically see something that looks like this S-shaped curve. And so in this case, the fluorescence of that sample is measured after the extension step at every cycle, and then it's plotted versus cycle. So in the early cycles, the fluorescence isn't high enough to get above baseline, so it's really pretty flat. And then as you start to build up product, you start to get into this exponential phase, and then eventually, as with conventional, you hit a plateau phase and you start to drop off, and you don't see a lot of additional amplification. The thing I want to point out here is that typically when you analyze data, and I'll briefly talk about it later, is there's a threshold set, typically by the software, but it can be user adjusted. And the point where the curve crosses that threshold is called the CQ value, which is either a quantification cycle, or sometimes it will be a CT value, which is the threshold cycle. So it's the same number, just given a different name. So the basic workflow for qPCR is you want to collect these amplification curves for a serial dilution of a DNA standard with a known concentration. You know, this might be a plasmid or some other double-stranded DNA that you know the concentration of, and then you collect it for the unknown sample, right? And then so the standard amplification curves, you see several there, and the one on the furthest left that comes up at the lowest cycle number has the highest initial concentration, right? And so you can think of it as the more you start with, the quicker you're going to get past that threshold, and you're going to get into that exponential phase. And the less you have, the longer it's going to take to get above that background fluorescence and start amplifying. So what you do with these is, once you have your amplification curves to your standard, you use that threshold and you figure out what the CQ number is for each of those. And they're typically non-integers, right? I just pulled up 20.37, you know, out of a hat. And so they aren't going to be even cycle numbers, right? They're not going to be 1, 5, you know, whatever. And so you take those values and you plot them versus the known number of copies in those wells, and then you can generate a standard curve. And then using the standard curve, you take your unknown sample amplification curve, and with that same threshold, that value that you use, fluorescence intensity that you use for your DNA standard, you use that, you figure out what the CQ is, and then from that you can directly determine the number of copies of sample in that unknown. So multiplexing is pretty straightforward, especially with the probe technology. All you need is a separate assay, which is a forward, reverse, and a probe. And the probe is going to have a different fluorophore depending on, you know, how many you need. So here's just a general example using a FAM and a hex. So this would be a duplex reaction. But you can go up to 5 colors with the CFX Opus. And how these are all detected is, I have a little cartoon on the right showing that there's a optic shuttle that traverses the plate and reads each individual well, right? And that's kind of how qPCR works. So then droplet digital was that next evolution of PCR. Same detection chemistries, right? I'm not going to spend much time on this. It's dye or probe. Typically, you'll see probe just because it makes multiplexing much easier. So in this case, the workflow is a little bit different, right? So this takes that PCR reaction and it partitions it using one of these disposable microfluidic chips to generate an emulsion with thousands of water and oil droplets, typically, you know, around 20,000. And so when you're preparing it, you put the generation oil in the oil wells, the sample in the sample wells, you put it into a droplet generator, and it uses a vacuum and the microfluidics to generate droplets. And then your droplets end up in that bottom well that's labeled droplets. And so in this sample, the DNA is just randomly encapsulated into droplets. Right? Some droplets are going to have no DNA. Some are going to have DNA you're not interested in, you know, just non-target background DNA. And other droplets will have, you know, at least one of the assay target that you're interested in. And so you end up with all these droplets, and then what you do with the droplets is you transfer them into a 96 well PCR plate, And then that plate is thermocycled to endpoint using a conventional thermocycler. And then removed, the plate is removed and put in a droplet reader. And the function of the droplet reader is to take the droplets in each individual well and singulate them, which you kind of see on the left, where you have this kind of this funnel with droplets, and they all get lined up so that they actually flow past the detector and the fluorescence gets red. And contrast to qPCR, which was using the optics unit that was moving. In this case, it's the droplets that are moving past the detector. And the little cartoon on the right shows droplets and intensity for like a duplex reaction. Right? One fluorophore is on top, one fluorophore is on bottom. You see some have green only, some have blue, and some have the green and blue. So what the data actually looks like is on the bottom here, right? So each accepted droplet is going to be plotted at fluorescence intensity versus droplet number. All right, and the droplet numbers are going to vary because while digital interrogates and looks at every single droplet that flows by the detector, only the best of the best fitting a strict QC criteria are going to be used for the concentration measure, right? And then analogous to qPCR, you use a threshold, and in this case, the threshold defines positive droplets above with a fluorescence intensity that's larger than the threshold, and a negative droplets that are below the threshold. And then what the software does is, in the background, it uses a Poisson algorithm and the droplets that you determined were positive and negative to directly determine your starting concentration. So it doesn't need a standard. It is actually counting DNA based on the partitioning. So multiplexing with this looks different. Typically, you're going to see 4 clusters. Here's a duplex data. Cluster 1 is going to be your double negatives that have no target of interest, no DNA or just background DNA. Cluster 2 is going to be, like in this case, FAM positive, which is going to have just the target for the assay that had the FAM probe. Correspondingly, 3 is going to be the one for hex. And number 4, you will most often see, unless you're at a very low concentration, is going to be a cluster that has both, right? At low enough concentrations, you aren't going to have both likely getting into the same droplet, but as you increase concentration, they're going to co-localize, and the software can take that into account when it's calculating concentration. So because of all that space, you can do advanced multiplexing and get even more out of a 2-channel, typically FAM and hex, right? So you can do what's called amplitude multiplexing, where everything kind of stays on axes, and you get all these individual clusters that are different populations of the targets. Or you can do a probe mix multiplexing, which kind of spreads them out and puts like that target 3 on the diagonal. Again, two different ways to achieve additional multiplexing over the available channels, right? And if you want to stay on axis, you can use a QX600, which has 6 fluorescent channels. In this case, FAM hex, Sci5, 5.5, ROX, and ADO. And just like with the 2D, the FAM hex, it is possible in this case to obtain up to 12 targets per well with advanced multiplexing. So you can, using these 6 fluorescent channels, you can get a lot of information if you need to. So the software, like I said, is really pretty straightforward. Here's like the example of a well. You can see your data in the top right as it shows the target and it shows your concentration in a tabular form. And then along the bottom, in this case, I'm looking at the concentration in a graphical form. And this just happens to be 6 targets on a plasmid. So you expect them all to be very similar concentrations, and they are. So it's just a good illustration of multiplexing, and this was done in 6 colors on axis. So to summarize the 3, you've got conventional PCR, right? Its endpoint is qualitative. It's kind of a yes or no, is it there? It's generally pretty low throughput, right? Typically you're in a single tube, or maybe you're using a tube strip, right? Or two. But again, you have to read those out through an agarose gel, and it is pretty low throughput. The qPCR is quantitative. It's detecting the fluorescence after each cycle. The concentration can be obtained, but it requires a standard curve, and can be very, very high throughput, right? You can get 384 well plates. Digital, again, is endpoint similar to conventional, but in this case it's quantitative because of the sample partitioning into these individual little droplets, and as a result of that, the concentration is directly obtained by Poisson analysis. And I consider it high throughput. 96 is pretty good. It's not 384, but you can still do quite a bit with a 96 well plate. So applications are expanding. And these are just some applications, right? Oncology is really taking off recently. But like you can see here, these are just some of the applications. The first part of this looked at water testing, and that was actually coastal water. Typically, people are using ddPCR for wastewater testing, and it was interesting to see that the coastal water was being monitored by digital, and they were looking for, contamination. The next subject of this is going to be cell and gene therapy. And again, there's numerous things you can do with digital. And then just kind of as an introduction to recombinant AAV, which is like really the main vector for human gene therapy. You get really great gene expression, and there's really no significant immune response or toxicity, right? And the dosage that they're using for these are based on the physical titer, which is the genome concentration, right? And so you'll see it in some units, some genome copies per mil or viral genomes per mil. So since the physical titer, the genome concentration is being used as a dosing unit, and you want to get reproducible experiments between preclinical and clinical and different labs to understand safety and efficacy, you really want to know what that physical titer is very accurately and confidently. And so it's typically measured by qPCR or digital, right? And then so I just threw this slide up here. I'm not going to walk through it. It's for your use of additional information. It is in no real order, right? The applications guide, which is down towards the bottom, is getting older now, but it's something I refer to quite often. It actually has really good background sections that kind of go over the math of Poisson and have some nice things you can look at and think about. The digital PCR methods and protocol methods in molecular biology, the first 3 or 4 chapters on that are also really pretty good background. They're a bit more detailed, and some of the benefits of digital. And then webinars, if you've got an hour, right? There's a bunch of webinars on the website, and they actually have what masterclass webinars, which are like these little 5-minute videos that I just typically just Google ddPCR masterclass or whatever, and you can find them, and they're really these nice short little videos that are really focused on one particular topic. So there's a ton of stuff out there, and that is it for me. So thank you for your time. If there's, I guess, any questions, I'll take some now, or you can wait till the end.
All right. Thank you, Andy. Any questions from the audience? I think Damon and Beth are going to lead the Q&A.
So if there aren't any questions right off the top of anyone's head, I can kind of get started with some general ideas of how to bring kind of some of these concepts into the classroom. So I predominantly have worked with a lot of high schools in bringing, PCR in, but I have a handful that bring real-time PCR in. I interact with a number of colleges and small universities that are also bringing it in. And so to some degree, it comes down to a little bit of what your pipeline is focused at. But I think the biggest thing that I see across all of them is making sure that they have like a very solid fundamental understanding of PCR kind of inside and out. Because if you've got that down, it's really easy for them to jump to the theory around real-time PCR, quantitative PCR, and droplet digital. And so I see a lot of classrooms will do this through animation if they don't have access to instrumentation. If they've got access to instrumentation, using some of the available kits to make it easier on you as the instructor is huge, because PCR, right, there's a lot of movable components and you don't want to spend all of your time quality controlling them if something goes wrong. So it's really nice to kind of work with something off the shelf. And then I have a handful of folks that will take that and extend it a little bit further towards real-time PCR by taking one of those off-the-shelf PCR kits. And then instead of having every student station run a full set, consolidate it down to 2 or 3 groups. And so what you can do is you can take all of those reagents, run them in large PCR volume reactions, and then sample them every 5 cycles and run those on a gel. And so they'll visually be able to see kind of what's going on during a real-time reaction. And then those I actually get to see do real-time. Most of them are getting the instrumentation due to their pipelines feeding into places where students are going to be potentially using this in their jobs, or institutions that are looking at doing some sort of long-term project that involves real-time PCR. So I've seen a lot of kind of eDNA sort of projects come through the high schools and the community colleges when it comes to real-time. Beth, do you have anything you would like to throw in there?
Yeah, sure. Just to jump on what you said, we in our qPCR and ddPCR class that we focus on those techniques, we actually will do gene expression studies. We've done it on whole mRNA from mouse organs. And then right now we are using C. elegans, and we do what we call a little quick crack method, where we'll use liquid nitrogen because we have access to it on site, but we'll use extreme cold and heat temperatures back and forth a few times to break open that cuticle of the worm and be able to access the mRNA. And we still do a separate reverse transcription step because we like to show the step, actually do the work, do the pieces of the work to do the reverse transcription to end up with your cDNA mix. And then we move into qPCR. And one of the best parts about it is that, not only are we doing something that's real, an application, that our students, our graduates might actually, encounter in their positions, but it gives students a sense of completion of, up to that point, we work them through labs that are pretty predictable, even though we're doing a lot of work that we're even using our students to test, future kits and future setups. But in qPCR, we really get to take something from beginning to end, work through all steps, and present at the end. That has been a great way to show exactly why qPCR and now ddPCR are such used techniques.
All right. Thank you both. And there'll be time for more questions. And if you have just basic questions about the technologies, please just go ahead and ask if you're just wondering, what is that? I mean, I didn't understand when Andy was talking about the very complicated multiplexing with 2 channels. That was a little bit much for me. But so if you want more information on that, just throw that into the chat. But we'll move on for this session. And I would like to introduce Claire Guion. Claire has more than 15 years of experience in a variety of scientific industry roles. She started her career as an independent toxicology laboratory in Indianapolis, Indiana, where she spent 8 years in quality control. She then joined Andelyn Biosciences in the quality control group before moving into analytical development. She spent the past few years specializing in the development of digital PCR assays and is currently one of the subject matter experts in digital PCR for Andelyn. And let's remember that Maranda is also here from Andelyn, who is one of their talent development folks, and can also talk to us a little bit about careers at contract development and manufacturing organizations like Andelyn. Thanks a lot, Claire.
So I just want to say thanks for having me today. Let me get this started. We'll go a little bit into kind of my career path because it is a bit unconventional. So as you can see, I titled this The Somewhat Accidental Scientist. I actually, I started my career in a bit different route, but here's a bit of a background on me. I am originally from Indianapolis, Indiana. I wanted to be a paleontologist when I was a kid because I thought dinosaurs were cool. I have some hobbies that are in the sports realm, especially being from Indy. I am a big motor racing fan. And I always liked science as a kid, but it was never something that I thought I was actually going to do. So my actual educational background is I've got a bachelor's in biology from Ball State. My original plan was to actually major in music performance. I was dead set that I was going to play the flute in a classical setting, and that was going to be my job. But that changed, and now my transcript has a ton of music classes that I don't do anything with. But as my kind of bio said, I spent 8 years in quality control at a toxicology lab where we did compliance monitoring, some forensic testing. So I helped make all the controls for that. And then I moved to Columbus, Ohio in the fall of 2016, where I started in quality control, kind of the same type of job I had before. And then I transitioned to analytical development a few years ago. So why science? I was accepted as a music performance minor. I had to figure out something to do. So I decided to go with the science realm, and then ended up dropping the music minor because it put me behind. I spent a lot of time in microbiology classes, but would have had to stay an extra semester for one class to get the microbiology focus. So I just graduated with a general biology degree. And even after all of that, I had no plan. I didn't know what I was going to do. I just said, well, we'll figure it out. So after 15 years, or actually after 8 years, I ended up at Andelyn. And currently what I do, I am a senior scientist in AD. There are 3 teams in AD. We've got basically the research QC side of things. We've got a qualification validation team and development. And I'm on the development team. And as I like to say, it's redundant. And, because you say I'm on the analytical development team, it's a mouthful. So I am responsible for a lot of development work for new assays, for method improvement. I do a lot of troubleshooting. A lot of my time is spent troubleshooting and helping other groups with their troubleshooting efforts. And I do a decent amount of technical writing. So all of my development projects, I'm responsible for writing our actual reports, a lot of our assay methods and our guidance documents that will transition to other groups, and all of that. So my main focus since joining the analytical development team actually has been digital PCR. So, and even that was kind of accidental. The way that I went about that is my manager at the time was working by himself to try to develop, our first ddPCR assay. And I asked if I could help him. And, 5 years later, I'm now one of the people in our company that focuses a lot on digital PCR. So just to give you an overview on Andelyn, we are a cell and gene therapy CDMO. We started in as like a group at Nationwide Children's Hospital. It was kind of developed and grown there. We eventually spun out into our own team as Andelyn Biosciences, and we have since grown in the past couple of years to where we now have 3 facilities. I work at the development center, but we also have the corporate center and the plasmid center. So we've just got a whole lot of things going on right now. And then a little bit more of our history is things have been going on since, the early 90s. I started in 2016, like right before that second facility opened. I actually started just in time to help move our entire lab over to that new facility, and kind of helped with that. But yeah, so I think we've got a lot going on. We're growing every day. Everything's happening all at once, it seems. But I think we started to talk a little bit about this, but we primarily work with adeno-associated virus, so AAV. All of our gene therapy products are based on AAV, which is a very safe method of gene therapy. It doesn't cause symptomatic infection, and you can kind of make it do whatever you want. So that's kind of what we do. Our clients kind of tell us what they're targeting, what they want us to make for them, and that's what we make. We've got the quality control team that will do the testing to make sure that as it goes through the process that it's it's what they want it to be. And then you've got the research side of things that are kind of helping innovate and figure out new and more efficient ways of doing things. But yeah, the AAV is a pretty interesting thing. So when I tell people that I work with virus, they don't know what to do with that. So yeah, so all right. So now that we've kind of gotten a little bit of that background, we'll get into a little bit more of what I do. And so PCR is a big deal at Andelyn. We use it a lot. We use it to determine our genomic titer of our samples. Traditionally in the industry, qPCR has been the methodology. That's what people are used to. That's what they know. But the current trend in the industry is to actually switch over to some form of digital PCR. It's tend to be seen as more, more accurate, more reliable, because it is an absolute quantification versus that, that standard curve based quantification. So we use a lot of PCR just in general across the company. And then you can also use it for, instead of just figuring out the genomic titer, you can also use it for residual assays. For example, host cell DNA. When we actually make our virus, we actually produce it inside human cells. So we have to make sure that the DNA from that actual host cell is removed as much as we possibly can. So we are trying to figure out how much is still there. And we'll get into that a little bit later. But here's kind of a sense of why we need to use digital PCR or PCR in general for our field. The FDA is very, very, strict in their, you need to know what your product is doing. You need to know, how much of your product is there. The dose determination assay is the only assay the FDA actually requires to be qualified. And that means that you don't just develop it, you actually test it with different parameters, and you make sure that it is as robust as you can make it. And then they also recommend that each of our clients have a product-specific genomic titer assay. And that means that we actually cater our assay to their product, which is generally using that. So that probe that was talked about in Andrew's presentation, we have a primer probe set that is specifically for that client's product. And then something that I do is when we are developing that and when we are optimizing, we actually have to optimize that primer probe set to make sure that it is being as efficient as possible. So the way that you do that is you would run a temperature gradient. So you would cast a wide net and to see what it does, and it'll look like this. So you're looking at kind of how your sample is going to come out of the instrument, and you are looking for as much separation as possible, and you're looking to minimize that rain, which is that area in between that top positive band and that lower negative band. You want that to be as clean as possible, but sometimes based on the sample that you have, it's not super possible, but you kind of do what you can. So once you kind of cast your wide net, you kind of look at what you think is a better, like, smaller range. So again, with that idea of maximum separation, minimal rain, and then you choose that smaller net, and you'll get something that looks like this. So a lot of the time you'll have this kind of result, and nothing really seems to be much different. So at this point, you just kind of choose one that looks good and you just go with it. They're all going to be about the same. It's just kind of what you're seeing at the time that you are actually looking at this assay. And so these graphs are actually from the development project that I worked on for our host cell DNA, like our residual host cell DNA assay. So this was something that I did. This was actually, I think, experiments that I ran to kind of determine, the very beginning of our assay development. So, because once you determine that, then you can continue on with your other experiments and really finish out the assay. But yeah, but even in science, it never goes right. So I spend a lot of my time troubleshooting, and I think that's a thing that for us, troubleshooting is something that we just have to know how to do. You get really, really proficient at it as time goes on because you have to do it so much. I think for us, like, we always start with that. We always double check our calculations. We always double check those little things, because so much of the time it's those little things that we just forgot, or something just didn't happen the way that we really expected it to. I had an example where I thought I grabbed the correct solution, and I actually didn't, and I got crazy data, and we had to go back and actually reevaluate a lot of things based off of what happened. So you can have matrix effects that you did not even know were possible in your project, and that just happens. That is what it is. Science is going to science, and you go with it as best you can. Yeah, and I think for me, I love teaching. I love having, bringing in interns and kind of like helping them really figure out where they're going, what they're doing. So I think for me, any like final thoughts or advice that I have is to take students through troubleshooting exercises. For me, it's like, what could go wrong? start to determine what that issue is. You know, kind of go from like the simple explanations to the more complex, because most of the time it's those simple explanations that really are the issue. It can be the more complex, like super scientific, we don't know what's happening. There are many times that we have tried to troubleshoot things and the data is telling us one thing, you know, but we are sitting here saying, but this doesn't make scientific sense to us, but we have data that is reproducible and it's just, it's going to do what it wants to do. You know, and I think that's, for me, you know, I didn't have like the highest grades in college. I didn't even, as I said, I didn't even have a full plan. I think there are sometimes, you know, our students or even our interns come in thinking that they need to have like a 10-15 year plan, like they have to do exactly this and this and this. That's great. If they're motivated, if they have this very good idea of what they want to do, that's awesome. For me, I didn't have that. I also did not figure out what my strengths were until I got into a role that honestly didn't even exist yet. We didn't have an analytical development team when I started to actually do analytical development work. And that's when I kind of figured out my passion and my expertise and the things that I'm actually really good at. I really like data. I really like data mining. I like trending and all of that. So, and I didn't know that that was really part of my makeup until I got a decade into my career. So for us, especially as an organization, we like to see candidates that have some knowledge, even if it's just like base, you know, like theory. They may not necessarily have needed to run the assays or to run the technology, but as long as they have basic scientific backgrounds and understanding, that goes a long way for us. Things change rapidly. Things have changed rapidly, even in the 7 years that I've been at Andelyn. So that's kind of, you know, it just keeping up to date on that. And yeah, and just like that last point of, you know, for us, if a candidate, if a student is eager to learn, that's really what we look for, because I can teach people to pipette, I can teach people lab technique, but if they're not motivated, there's not much I can do. So for us, it's kind of looking at that. Do you have that willingness, that eagerness to learn, and just kind of go with what's happening in science, and to understand that things are not always going to go the way that you planned, and for the most part, they're never going to go the way you planned. And, you know, are you flexible enough to kind of go with that? So I think that's all I have. So I can end there. You know, again, thank you for having me. I've enjoyed preparing for this opportunity. But yeah.
Thank you so much, Claire. And Maranda, if you want to come on for a second, I'd love for you to talk about the types of jobs that are in Andelyn and the types of people that you are looking for, especially, you know, roles that are difficult to find that might be coming from these instructors here. That would be great.
Yeah, great job, Claire. You did wonderful, and thanks for having us again. We really appreciate the opportunity and the platform to share. As Claire mentioned, we're Andelyn Biosciences, so we're a contract development and manufacturing organization. And with that, we have a lot of specialties within our group, not only analytical development, but we also have operations that is actually manufacturing the client that goes into humans, research level work that goes on. So we kind of have two different pathways, more of this technical level where we have scientists ranging from technicians all the way through senior scientists, principal scientists. And then we also have kind of the specialist track, which can be more of our quality assurance type roles through all of our business avenues as well. So human resources, IT, et cetera. But for more of these technical roles or positions, like Claire was alluding to, we really seek out self-motivated, and I think Claire hit on some of these points already, but just self-motivation, self-starter, eagerness to learn. A lot of our groups have areas of needs or roles and positions where, yes, it's great to have like a strong microbiology background or something, but as long as there's that eagerness to learn and willingness to come into our organization, our teams have great training programs where we can kind of really build these skills into, and I know our teams really value that ability. Also, some key things, just being a contract development manufacturing organization, very important to be quality minded. Quality is everything. So we really need to focus on that. So just making sure that students kind of understand that quality mindset, what that means into an organization, because the products that we are manufacturing do touch patients. These are human use products that will go into clinical trials eventually. So keeping that in mind, as well as good documentation practices, very essential for our organization. So just kind of building in those key concepts. I know a lot of lab work goes on, but kind of, enhancing, some of those thoughts and processes and how that can transition to industry, like Andelyn, which is really important. And then just being very detailed oriented as well is really important with any of our roles within Andelyn. What we do is very important. A lot of the work that we do, we have to keep in mind that product that we manufacture, produce, every biome has a name on it essentially at the end of the day. So we really value individuals that come into our organization, eager to learn, self-motivated, and really want to make a difference. And that is our vision and mission as an organization. So hopefully that gives you a little bit of insights. If there's specific questions, more than happy to answer or address those.
Thanks a lot, Maranda. That's a lot, and it sounds like it's a fun place to work. Claire's smile shows that too.
Yeah,
I will give the floor over to Damon and Beth to talk to what we just heard and answer any questions that come up in the chat. So if you have questions, please throw them in the chat.
And actually, just over there in the chat, there is a discussion going on about RNA isolation kits. And so if anybody has any other feedback that they would like to give that's been doing that, please feel free to jump in there as we're trying to throw options out. I think one of the things that kind of like stands out to me there is within this context as an educator, it's really important to show these different career paths for students, because it's really easy, right, to think that like, I only have this path in life to get to this one career I've heard about, where most people like Claire and like myself, by the time you're actually into your career and you look back, you're like, wow, that was not a straight path or what I anticipated. And so I think anytime you have a chance to share those experiences with students is really important. And so that's why I think getting guest speakers in to do that a bit that are, from industry locally and things like that are some of the biggest things you can get. Get them in there to talk about what's going on in their industry, but make sure they get that career talk like in the first, 5-10 minutes of it, because I think that really helps give students perspective.
I would highly agree with that. We seek out, we ask just about every guest speaker we bring in for whatever reason to give a snapshot of how they ended up where they are, and most of the stories do not start out as, I earned my bachelor's in a bioscience, went on to a PhD, taught for a while, and then started my own company. That's rare. We have people from all walks of life. I started as IT. I started in IT and needed a new life and landed in our bioscience, our biotechnology program. But we work with a lot of different people and a lot of different types of students, and knowing that or getting across to our students that it's not all bench work. You're not going to sit there and work at the bench or even, write reports and present all of the time. And you don't even ever have to do that and have a successful and rewarding career. Do you like computers? Do you like data? Well, maybe bioinformatics, data science is your path. Are you a people person? Do you like talking to people? Do you like marketing? Then we have students who their bench skills, we work with them a lot, but they're just not stellar by the time they end up at the end of graduating. But we've got a couple that they went on to start their own customer service management and marketing firm to work with local startup companies in the bioscience, biotechnology arena. You know, HR, are you a perfectionist? You know, then maybe a QC path is the way for you to go. There's not just one thing that you do in biotech. It's not just choosing animal, plant, or human health. You know, it's not simple buckets anymore. You've got the wide range of careers that a good basis in biotech will put you on a path to success.
And inside of that too, I think there's anytime you have the chance to kind of bake in soft skills, because it's not something that gets, you know, just pointed out in a lot of coursework, right? And anytime you get a chance to build that in, it makes for much stronger employees down the road when they hit their careers. And I've seen this in the different schools I've interacted with. I've got a handful that actually that is a large portion of their curriculum of every unit. Is at the end of it, the way they do the share out with the class, and they have to do that to externals, which is huge. And those students, I feel, when I talk with them, they feel like they're 2 or 3 years ahead of every other student at that age that I talk with. So anytime you can also make sure that you're getting soft skills kind of baked into your units also is very, very helpful at kind of developing that. And in that process, what they'll realize is that, oh, if I've got this skill set and I understand the science, now I'm really qualified to go after these other roles within kind of biotech industry, which, as Beth points out, it's, you know, it's a small percentage actually that are in those lab coats at the bench every day. And there's a lot of people that have to understand the theory, but then have all these other skill sets that help make the organizations work.
We had a question about if we could explain what rain is and what are common causes of seeing rain. I've got some ideas, but you know, I'm not the expert. We got them on the call now.
Andy, you want to take that one?
Yeah, sure. So rain is what we call like intermediate intensity fluorescence. So in a clean experiment, you're going to have like the ideal 1D that I showed. You're going to have a negative population and you're going to have a really large positive population, right? But there's a variety of things that can cause rain, and rain is basically just droplets that didn't have quite as efficient amplification, right? So it didn't chew up a lot of probe and push the amplitude all the way up to that top. And so, you know, you would like there to be none, but there is some, and it depends on assay, and it varies from assay to assay. Typically, one of the big causes is just accessibility of target. Right, is like if your primer probe can't get to that target, it's not going to amplify it very efficiently. So it's going to kind of lag behind several cycles until you get enough of the small PCR product that can use as a template, and then it really takes off. So that's kind of my take on that.
When we work with CRO partners or industry clients with ddPCR, that's usually the first culprit that we start identifying or looking at. And if they're working especially with genomic template, then digest some more. Do some more digest, make that fragment, that section that you're looking for, more accessible. That's the first one we go for. We had another question about cost differential between ddPCR and qPCR once you have the hardware. And then also sensitivity of ddPCR versus qPCR.
I'll try to jump in again.
Thanks, Andrew.
So I don't have the exact numbers. I believe qPCR is a little bit cheaper per well than droplet. But so, and then on that other question, droplet is more sensitive. It's less prone to inhibitor issues because you're partitioning and kind of diluting out the whatever interfering substance may be, so that within each droplet you may not have that inhibitor. And it can go low. I mean, you can get like 5 copies per microliter with droplet, and so it will work in the low range, and then it goes up to once you start running out of negative droplets, droplet doesn't work anymore because you need to have positive and negative. So the upper range for droplet is in the 100 to 120,000 copies per microliter. And then doing the math with that, if you only have 20,000 droplets, there's going to be many multiples in those droplets, right? But Poisson takes care of that, and so the range is from very, very low up to about 100, 120,000 copies per microliter.
So we had another question about implementing ddPCR in the classroom. How did you break up your students, the number of students per group, for instance? And are there any available resources for projects that can be implemented in the classroom? Referencing that worms example that I mentioned. Damon, do you have ideas for that?
The only one that I've really kind of bumped into was a group that was looking at doing kind of some environmental monitoring using ddPCR, just because it's got that lower sensitivity. But I haven't had a whole bunch of hands-on experience with bringing those in the classroom at this point.
And I think this is where the hands-on workshop would be great. Xiomara, if you're free for that next year, that's where Beth and Betsy will be talking through the things that they do. And I know that they're happy to share the things that they do in their classrooms with people as well. If you want to reach out to Beth and Betsy, who are, I'd say, leaders in this right now, because there's not many people doing digital PCR in the classroom. That's why we're doing this webinar series, to kind of say, this is something people should start to teach. We in the Explorer program have the kits to help you start real time. We haven't got to Droplet Digital yet because teachers are only just starting to do it. But we're starting that process as we move forward. This is part of starting to introduce these concepts.
One thing I picked up on that I wanted to go back to, I think it was Claire who was talking about, I know, troubleshooting, but in qPCR and definitely in ddPCR, in droplet digital PCR, other than pipetting technique, and I joke that qPCR and ddPCR expose your pipetting technique, especially with learners. You can go through a lot of training and think you've got great technique, and you do, until you get to qPCR and ddPCR. And then you find that it's extremely important to be consistent and, to have your own methods. But troubleshooting skills are key. It is more worthwhile to know how to troubleshoot because you have a good understanding of the technique, what's going on in those wells, and the science, the biology of what you're working with, because being able to troubleshoot, again, is almost more valuable than being able to perform the technique. Having those critical thinking skills, applying those to something like figuring out why something went wrong, and even better, what are the options to moving forward? It's everything to the employers that are around us. Going to pipetting technique also, I think Linnea in the chat mentioned about working with students to create master mixes as opposed to small volume pipetting. That is the very first thing that we cover, that we hit on in our classes. And even when we're talking to students that aren't in our qPCR specific class, that anything to illustrate the issues with low volume pipetting is great. We'll use like 96 well plates or even, fewer well plates, and have students, in the first column, maybe they pipette a microliter or even less. In the second column, they'll pipette 10 microliters and so on across whatever the volumes are. But at each point, each time we have them do a column, we'll have them weigh the plate. So they'll, weigh the plate empty to tare it, and then weigh after each column and see how close to theoretical they can get and what their error is. That is often eye-opening to students. They think that they're doing exactly right to pipette, a microliter or even less. And more often than not, it's revealed that there's extremely high error rates there, at least when they start.
And another way to do that is also just with, if you've got a spectrophotometer, doing that with a dye, and that way they get to build the standard curves and things like that. But yeah, it definitely reinforces those pipetting skills really quickly. And the nice thing is with that, it's often one of those ones that you can appeal to students' kind of built-in competitivism as well, because then you can be like, who can give, the best standard curve at the end of this, within this set of pipettes. The troubleshooting, though, I think is really, really key, but that's something that you can start really early on in your programs too, when you first introduce PCR. If you're doing PCR labs where you've got a lot of appropriate controls built in, I like to go through and even just dummy up gels of all the failure modes, and then spend a lot of time at the end going through the different failure modes and having them diagnose those. So actually in the chat, what I'm going to do is I'm going to drop one of the Bio-Rad kits. The GMO kit is actually really great for this, because you're starting with food to begin with. They're grinding up, extracting the DNA from, but then it's got a lot of appropriate controls built in there. So the end of this slide deck that I just put in the chat has all those different type of gel phenotypes that you might get, and you can have them go through and try and diagnose what's going on. It helps get those skills going early, so that way when you get to real time, they've got that built in, and they're just adding on the next level of knowledge about how real time can have issues that come up that are different.
I'd be interested to hear from Claire. Do you use formal troubleshooting methodologies like failure mode analysis and things like that? Or is that just in the manufacturing group that does those kinds of things? Fishbone diagrams, all those things, and how important is it that people know about those things from an education perspective, and students coming in to understand that there are formal ways to troubleshoot versus just kind of working through what you think went wrong?
Yeah, I think we do have some we've done some formal troubleshooting in the past, at least in analytical development. It's usually experiment to experiment. So we tend to sit down and go through, okay, what's actually happening? Sometimes it's a formal type of troubleshooting event. Sometimes it's just the analytical development team sitting around going, what happened? We don't know, kind of going through step by step the experiment and trying to make sure that what we did is matching the data that we got. And yeah, because I've definitely used some formal troubleshooting tools and techniques. But yeah, I think for us, it tends to be more towards that critical thinking, like creative side of what could possibly go wrong here? Why are we seeing what's happening? And kind of like backtracking what we got.
And I'd be interested in documentation and the importance of that in your work, because I can imagine you're making things for FDA that's a big part of what you have to do.
Documentation is huge. Even in our development team, you still need to make sure that you're documenting well and legibly and clearly, so that people come back and see what you did, how did you develop the assay? Because yeah, that you can actually, like the FDA can come in and say, please show us exactly how you developed this assay. And if I did not document well, if you didn't write it down, it didn't happen. So for us, especially with our methods going into the actual manufacturing side of things, from the research side into the manufacturing side, we need to be very clear about what we did, and documentation is it is a skill that in all reality some people struggle with. We've had to really have some workshops on, okay, how do you document properly? How do you correct documentation properly? And all of that. So yeah, that's something that it's an ongoing process, but it's a big deal.
It is a key component of our program too. And I know I keep referencing our community college program, but that pretty much underlies everything. It's one of the first skills that we try to get across, to the point where I don't even think about it and mention it, because it's just a given that you must. There was, I think we didn't address, for those of us that implemented ddPCR in the classroom, how did you break up your students in terms of number of students, in a group? Damon, I don't know. I know that ddPCR really hasn't been pushed out into the classrooms that much. I don't know if you have any experience with that, but typically we try to keep our groups at one or two in a group. Two is nice, especially when you're doing introduction, because no one feels that everything is just on them. And it's always nice to have someone else watching, help you document while they document. It's nice to do that. But beyond two, maybe three, then you're going to have people that are standing in the back and not doing anything.
I completely agree with that statement. With real time, you can stretch that a little bit more, usually because you've got more samples that they're processing. So each group then can be a little bit larger because everybody's going to have a chance to process samples. But with ddPCR, I think, yeah, two is a good number.
We have a question. Does Bio-Rad have a basic qPCR kit to look at the gene expression of a housekeeping gene? I think you're on mute, Damon.
I just answered that in the chat. We don't have one for gene expression at the moment. We only have two kits that do real-time PCR, and they're built off of our existing general PCR kit. So it's one that takes the GMO kit and turns it into a real-time experience, and then one that takes the CSI kit, the kind of the crime scene investigator one. From working with colleges and unis, I find that the GMO one is a much bigger kind of learning experience for them because there's more built-in controls. Just the overall experience, I feel like that one is the one I would usually go with if you're looking for an off-the-shelf option right now.
Yeah, thanks, Damien. I did see that on your website. Thanks.
Yep.
And then Jeremy, you asked how many times does a student go through a PCR exercise during the program? Are you asking about our program specifically over at SDLCC? If so, general PCR in the 2-year program or even the 1-year certificate program, I don't know how many times, we go through PCR. qPCR and ddPCR are mostly right now limited to our qPCR focused class and then our forensics techniques focused class. In the forensics side, we use qPCR right now to quantitate the, we have students use swabs to take a buccal sample and then extract their DNA. And then we do a human ALU assay, which isn't really a pretty assay. You don't get a nice, strong single peak on the melt curve when we're doing a cyber type assay, but it's enough that, you get 2 peaks or so and, you're good. We use that to quantitate the amount of template we need to put in for our fragment analysis for the human ID, human genotyping assays. And then of course our qPCR class. And, that's either an 8-week or a 16-week course. So we start at pipetting technique. We move through comparing if you pipette components individually versus making a master mix, and then we go from there. So I don't really have a number for you. I'm sorry. PCR, a lot, countless. qPCR, it just depends on what special topic courses or what we call them.
I had a more question about AAV, and are there any actual diagnostics or no, sorry, clinical, is any AAV through clinical trials yet? Is there anything like in the clinic yet in clinical with AAV? What is it being used to treat?
So it's actually being used to treat a lot. I believe there, if I remember correctly, I think there are there 5 that have, there are 5 that have actually gone through full FDA approval. So there are like commercially available AAV based gene therapy products out there. There are a ton still in trials. It's a growing industry, and it's I don't know how many are on the horizon, but I know that there are still some in the works for approval. So, but yeah, no, 5 have been fully approved by the FDA at this point.
I think it's notable to call out what makes AV so special is you can typically get 1 dose or a couple doses, and it's meant to really treat or even be curative. Like Andelyn, we're named after 2 gene therapy pioneers, and the second half of our name is Evelyn Biosciences, and she received a clinical trial product that was done at Nationwide Children's Hospital that we had a hand in manufacturing. And that product went on to be FDA approved, called Zolgensma. It's one of the most expensive drug products in the world right now. But the great thing about it is that you get 1 dose. And Evelyn received that when she was just a couple months old. And now she's, I believe, 7 or 8, running and walking and living her best life, where she was diagnosed with SMA type 1, which is just absolutely devastating and robs her ability to move, breathe. So it's pretty fascinating work that we're able to do and partner with our clients on. It's very rewarding.
Great. It's so exciting that the technology is moving so fast, right? That someone 7 years old, and that's the time Claire's been with the company.
Yeah, no, I think she was dosed like the year before I started. So yeah, she's basically been part of the story for about the same amount of time I've been here. It's been a really, really cool thing to be a part of.
Awesome.
Yeah, we constantly talk about how technology seems to be moving faster and faster as time goes on, but in something like these advanced PCR techniques, you can see it, in the relatively small amount of time that I've been on the bioscience side. We went from qPCR being, a standard, and ddPCR being something that people were still thinking about and working on. So it's pretty incredible.
I actually have and Andy maybe can correct me if I'm wrong, I'm not sure, but I think that Bio-Rad has an SMA test for droplet digital, and it has controls in it. That's actually one of the ones that we thought, and we haven't tested it yet, but we thought would actually be an example that teachers could use because it has the controls in the kit. And I think we have to check that, but that's a nice story if we've got the Andelyn story, and then we actually have a little ddPCR assay to test for the presence of SMA, because I remember having going to a talk where the people that have been working on this were talking about it, and they collaborated with Bio-Rad. So I'll need to check on that, but I think that we have that as a product, and it's got the control. So you don't need a sample from a patient. You have the control, but you can run that assay through a ddPCR. Right, any other questions? Or I know that Cassandra would like everyone to do the InnovATEBIO survey, which she's dropped in the chat, and I'm sure she'll drop in again. In order to incentivize you for that, you can get a free poster from Bio-Rad. When you fill that in, there's an option to click a link to go and get a free poster. Those surveys are really important. InnovATEBIO is funded by an NSF ATE grant. And that is funding the presence, the ability of us to bring you this. So please help InnovATEBIO by filling out the survey about, your experience here and help them get better. And then Jeremy asks, what comes next after ddPCR? If anyone would like to take that one. Andy's shaking his head saying, no, this is it. We're at the ultimate now.
No, I mean, you got the best. Why move on? I'm sure there's a fourth generation out there, but we're still, working through the third generation, which really kind of only came around in like 2010 or 2011, I believe, is when it really started being commercially available. So it's still, and on the time scale of things, it's still a relatively new technique. But yeah, I'm sure there's a fourth generation.
I hope you don't mind, but I just want to say I couldn't believe how many of these machines were at Fuji Films contract manufacturing. It was like a Bio-Rad ad.
Awesome. That's great. Bio-Rad is leading the field in droplet digital right now, so it's really nice to be part of that, so to watch that happen.
Yes, and please take the survey because we want to do more of these things. These type of webinars followed by face-to-face workshops.
And Cassandra, there's a question about how many details, about getting details on the hands-on
Yes,
PCR workshop.
we will be sending out more details probably in the January newsletter for InnovATEBIO. So become a member of InnovATEBIO and sign up for that, and you'll get more information then.
Thank you for Cassandra to reminding individuals to sign up. It is a free membership, but then you're part of our database for sending out information on the availability of webinars and workshops.
All right, I would like to say thank you to everyone, to Claire, to Andy, to Damon, to Beth, and for everyone else, Cassandra, Dion, Linnea, Purnima, Ingrid, for helping put this on, and Maranda, who I have not forgotten. Thank you so much for helping to coordinate as well, Maranda. Maranda came to the InnovATEBIO Summit this summer. Andelyn's interested in really supporting workforce development in this area. And so that's where we met Maranda today, for today. Thank you very much for coming.
Yes, and I echo that thanks, and happy holidays.
Happy holidays. Enjoy next